Who wants to ask the first question? Manu.
This was a great session, Mark.
Thank you.
The first question is to Paloma. In terms of she had a wonderful slide on the chemical modifications of aptamers, pointing out the O-methyl, fluoro, and amino. So the question is, of course, the chemical toolbox is much beyond that. And what is the acceptability of these aptamers at present so that we can have reasonable size aptamers for a wide variety of applications, and what can be expanded? And so can you please elaborate on these chemical modifications?
Yeah, man, is this on? Yeah, great question. The chemical modifications that are being applied to aptamers are very similar to all the other ones that are being used for other oligonucleotide therapeutics. So they are expanding. In terms of the ones I presented, those particular ones at the 2' position are the ones that we can easily incorporate during the in-Cellex process. And so we are limited, you're correct, in that the polymerases will not efficiently incorporate many other types of chemistries and modifications. So we're limited to those particular modifications during the in-Cellex process, and post-Cellex we can incorporate additional modifications to impart on these aptamers a lot of different types of properties that we're seeing with other types of oligonucleotides. So we're probably going to hear later on Tuesday from SomaLogic and all the different types of modifications, the hydrophobic modifications that they are using for their DNA aptamers to improve not only stability but also affinities, and also identify aptamers to targets that in the past have been almost impossible to target with this technology. So I don't know if that completely answered your questions, but I think we're there. Yeah.
Yeah.
Hello. My question today is for Jennifer. I was wondering, besides sequence homology, what other factors could influence the need to test in 2 species versus 1 preclinically?
That's really the primary one that we have to deal with as far as needing to consider whether we do 2 or 1.
Okay.
Did you have something in mind?
I was just wondering if there was other factors such as the lack of available treatments for a specific disease, for instance, or other considerations besides that.
Yeah, so typically we don't do the tox studies in, say, a disease model. I'm not sure if that's where you're going, but typically it is done in the healthy animals.
Okay, thanks.
Morning, everyone. 2 questions, if I may. One for Mark. I mean, you commented on your expectations of there being, let's say, relatively modest growth in manufacturing capacity. Could you maybe briefly also comment on what you think is happening on the feedstock side in terms of supplying mainly amidites, I guess, to CMOs? Are you seeing any, significant dynamics there? And one question also on toxicology. You didn't mention carcinogenicity studies in your presentation. I guess, again, here, ICHS6 might be interesting in terms of whether or not that is required or recommended in the oligo field. Thanks.
Oops. Okay, thank you. For the first question relating to the safety of supply of amidites in a world where more oligonucleotides are necessary, that's something that has been investigated for quite some years now. And so far, based on a lot of different indications, including some visits I personally made in different facilities in different countries, producing not the amidites, but the precursor, those nucleosides that are used and transformed into phosphoramidites, the fact that huge amounts of nucleoside inhibitors of virus replications is made, AZT to start with, and other products in the same class of molecules, needing tens of tons of those nucleosides. And the fact that everybody is now relying on synthetic nucleosides for making amidites makes the supply relatively safe. And we have discussed some projections for the need of hundreds of kilos of oligonucleotides, meaning that we need many tons of those phosphoramidites. It's pretty much under control. Of course, some capacity increase will need to be implemented, but they are known. People know how to handle that. It's just a question of months to build up larger synthesizers or larger columns for purification. It doesn't seem to be a real issue at the moment. And for solvents, because it's also a question that has been addressed, we all remember the 2007-8 kind of shortage in acetonitrile. And since that, companies have developed plants making acetonitrile for the sake of making acetonitrile. Not only as it was in the past, where acetonitrile was a recycling of a waste coming from the production of acrylonitrile. But people now are making acetonitrile as a real product. And in different parts of the world, those capacity to make acetonitrile have increased, and it's also generally admitted that it's not a real issue for the coming 3, 4, 5 years. And if some products would become ton products, we have time to increase the capacity. So it's under control.
All right, so back to the question about, I think it was carcinogenicity studies. So I was really focused on early stage programs here. So obviously, it wouldn't be done for an early stage program. I think just in general that we are going to be expected to be doing a lot of the late stage juvenile tox, embryo fetal. I think all of that is going to have to be done at some level with oligos.
Maybe a question for Marc, I guess. So as we move for a higher level of manufacturing, do you think that companies will be moving from solid state to solution phase synthesis of manufacturing? And do you have any comments on that?
It's a very interesting question. In theory, solution phase synthesis for oligonucleotides could be a good way to manage costs for projects where people would need to make metric tons of oligonucleotide. The type of equipment needed to make solution phase is generally significantly less expensive than what we need to make solid support synthesis. We need to address then the question of purity, of coupling efficiency we can achieve in those solution phase. I've been involved in some development in the past around solution phase. This experience indicated to me, and it's a personal view, that if something like that would develop, some sequences are probably better than others to deal with this solution phase. And the point is that based on what has been developed, published what we know today, it will require a significant development, so some investment. It's certainly not impossible, I guess, but it will take some time, and that's something that will need to be supported by some research program. Yeah.
Yes, I was wondering what the feelings are about nanoparticle encapsulated nucleic acids versus naked nucleic acids, and if you think it's time, given the success of the naked nucleic acids, that the nanoparticles would be reserved for messenger RNA or plasmids where you need to use them, or is there still a role going forward for new projects using nanoparticles for small oligos that could, in principle, be delivered naked?
Well, I didn't state it in the talk, but it was implied, and you just stated it well, that the lipid nanoparticle technology is certainly still attractive for large cargo, where the naked conjugate approach probably will not be successful. So I think that's a very good reason why research in those systems should continue. I certainly think it would be worth academic groups continuing to explore them for small cargo as well. But at the moment, the conjugate approaches, at least for hepatocyte delivery, seem to be more compelling.
All right. I also have a question for Paul. You just mentioned, if I understand correctly, that most of the formulated oligonucleotide drug product is still in aqueous form. To date, how much we know about the stability of these drug product candidates? And aiming for success beyond phase 2, how much effort do you think companies should put into developing a non-aqueous form formulations before entering into clinical study?
Are you referring to storage stability?
Yes.
Yeah, that's not actually an area that I have a lot of experience, but I think that the programs that are in the clinic clearly have sufficient stability to support their clinical development. The stability, of course, is going to be a strong function of the actual, chemistry. What are the chemical modifications? The Ionis portfolio, for example, I believe is all fully phosphorothioated. What the sugar modifications, base modifications happen to be would all impact the stability, especially the chemical stability.
All right, thank you. And the second question actually is for Jennifer, and good to see you again.
Before you ask the second question, I can also comment a little bit on the stability of the drug substance, the oligonucleotide itself, and some of the drug products. The oligonucleotides are not that unstable. So I have seen data showing that stored at -20 and even stored at 5 centigrades, many oligonucleotides, if not all, are stable for at least 24 months, sometimes up to 36 months. And for conjugates who are not formulated in a very specific way, just a lyophilized, sterile product, you have more or less the same kind of stability. So stability of an oligonucleotide is not really an issue. When you formulate that in LNPs or nanoparticles, then you add another dimension, that is what will be the stability of those structures over the time, and that could be a lot of different things, including peptides and what kind of interaction between the nanoparticle components and the oligonucleotides. And I think we don't have really a lot of experience on that, and that will be analyzed on a case-by-case study. But for the drug substance itself, siRNAs, synthetic siRNAs, extremely stable.
All right, thank you. And my question for Jennifer is, these days we know FDAs can be quite unpredictable. And so for oligonucleotide drug product, so they're largely referring still to, especially formulated in nanoparticles, referring to the liposome guidance. How flexible do you think they are in terms of changing the guidelines based on specific drug product? And to date, as far as I know, most of the QC release are still based on analytical assays. How likely do you think FDA in the future will require a biological assay to ensure the biological function reproducibility between batches?
I think there were two questions there. So I'm going to start with the first one. So yeah, there is a liposome guidance, and yes, I think the FDA does tend to pull that out, typically in comments when they've reviewed an IND. I think it's really up to us as sponsors to make the argument, for or against, depending on what they're suggesting. I think that's why some of these early dialogue opportunities with the regulatory agencies are so important. And I think we need to, show them the detail about our formulations and why this may or may not be applicable. Generally, I think it's not. And I don't typically use that as a guidance that I look at for putting together a LMP package. Right, I think I'm gonna hand over maybe to Mark, your question about release and use of, you're suggesting is there need to have a biological assay for release?
Yes,
okay.
Precisely.
I did not follow all the projects. What my understanding is that a biological assay could be important for oligonucleotides like aptamers, where you have a folding issue, just like for some proteins or antibodies. And as long as you do not understand and can say clearly that lyophilization will not modify the structure that could in turn modify the biological activity, it's nice to have a biological assay. For siRNAs, antisense, and other type of sequence-dependent products interacting with messenger RNA or something like that, I don't remember of any program where FDA has asked specifically for a functional assay as part of the release.
All right, thank you.
Two questions here. First one is for Jennifer, is from candidate selection to IND, you talked about two pathways, if you will. One is the general program and the other one is the streamlined one. In your experience, or even theoretically, what do you think is the best case scenario for the streamlined program?
How fast can you get an IND out the door?
Yeah.
Essentially. I would say for companies that this is not their first time out, if you're looking at a very short tox program, possibly you could do it in as little as a year. But that, everything has to work. Nothing can go wrong. No glitches in the manufacturing timeline, anything like that. And you'd have to have a very short tox program because, unfortunately, when you talk about doing a 1-month study, there's 3 months behind that as far as the analytical and getting the documentation put together in the reports. So it's a bit of a misnomer to call it a 1-month study because in reality it costs you, from a timeline standpoint, months, 4 months, 5 months. So yes, I think you could probably squeeze a program into a year. I typically say 18 months is a much more comfortable timeline, and my streamlined descriptions probably aren't going to take much more than, maybe a month off the way that I've described it. So hopefully that helps.
Yeah, thank you. And the second question is for Paul. How optimistic personally are you about extrahepatic delivery of oligonucleotides that are clinically viable?
By the systemic route or by the local route? By the systemic route. Well, I guess if I were, so I consult in the space, I don't have a lab, I don't have a pipeline. If I had a lab and a pipeline and a company, I would probably prioritize other opportunities. So I think that it's an area where there's room for an enormous contribution from the basic research side, which in the current climate I would say is probably going to have to come from the academic side. And I don't really see a lot. I see a lot of interesting ideas, but in terms of, and it's certainly something worth following. But in terms of things that are ready to fast track, for Jennifer's streamlined fast track, that's hard to come up with.
One of the recommendations we heard this morning from Marc Lemaitre was don't include modifications you don't need. I thought that was very interesting. And I would like to ask, especially regarding the 2' fluoro modification of RNA aptamers, what your thoughts are on a single pyrimidine 2' fluoro modification versus modifying both pyrimidines in an RNA aptamer. Are there any advantages or disadvantages to either option?
So modifying essentially all pyrimidines in the RNA aptamer sequence, is that correct? Normally, for the therapeutic aptamers, because we introduce these modifications during the Select process, we automatically modify all the pyrimidines within the sequence with the 2' fluoro. Several groups have also described the 2' methyl modification, again modifying all of the pyrimidines with the 2' methyl. What has been done a little bit differently is during the post-selection process that people have looked at modifying only one or several of the remaining unmodified purines within the sequence. And that even in our experience has made a big difference, where if you modify only a few of the purines, you don't actually get an increase in stability of the aptamer. But if you tend to modify more or all of them, then you see a great increase in stability of the sequence. So in general, I think with many, if not all, of the oligonucleotide-based drugs, I think the more modifications that you can have that do not disrupt the function of the oligonucleotide, the better.
And for siRNAs, I think it's important to analyze different parameters. On one hand, modifying your sequences can increase the chemical stability of your molecule. And it can also modify how long those molecules will stay in the cells, and that's the most important. So you will have to tweak your lead compounds, make this kind of medicinal chemistry comparisons. So if you can demonstrate, for instance, that by introducing some modifications into the siRNA, you can reduce dosages from once every week to once a month to once a quarter, or as some current clinical trials may suggest, maybe once every 6 months, the additional cost of those modifications will be paid off several times by the fact that you have to administrate much less product and just a few times a year. And all those parameters are very important. And then also don't forget when you plan some design to make a quick check that you're not going into some patent infringement in case.
My question's directed for Dr. Burke. Oftentimes, I liked your Venn diagram, chemistry, formulation, and biology. So on the biology side, target selection almost seems to be assumed, and then candidate selections where a lot of companies seem to focus. The target mRNA, cytoplasmic, we're going to target it with either siRNA or an ASO. But delivery and escape from the endosome still seems to be an issue. So the amount of the oligo that you can get to escape from the endosome then becomes critical. If you were to target something such as a link RNA in the nucleus or an enhancer RNA or something that is a low abundance transcript, you would think that you would need less ASO, for example, to do that, thus kind of circumventing one of the problems of endosomal escape. Have you seen a lot of companies moving towards trying to find these targets that might either up or down regulate a target gene, such as an enhancer RNA or a link RNA that's suppressive, or has the science not been there yet for them to really use that as a rationale to go after those targets?
Well, this is a great question. I think the short answer is the science isn't there yet. So I did refer to the enormous potency difference. I think it was around 10,000-fold between the siRNA liver levels and the mipomersen liver levels. And while the mipomersen is, I believe, is considered to be an RSH mechanism, which is catalytic, the turnover with the risk-based mechanism is, I mean, I'm assuming just much, much greater. And that accounts for the difference. So when it comes to the long non-coding RNA, the jury's out. You know, we just don't know yet. And that will certainly be an interesting parameter to monitor. And as you point out, it's highly relevant to the achievability of the delivery goal.
Yeah, Manu?
Mark, this has been a great workshop. I just want to add 2 comments to previous questions. One was based on the 2'-fluoropyrimidines. And I just wanted to mention to you so that everybody goes with the clear message about the 2'-fluoros. The 2'-fluoropyrimidines as well as purines have been used extensively now. And what we know, compared to some of the sharedness presented by the earlier literature and so on, those molecules are very safe, they are very well tolerated, they function greatly, and to this effect, there are going to be at least 2 posters by Alnylam, so which addresses this. So you can use it in aptamers, siRNAs, and everywhere. So that's one thing which I wanted to mention from our experience. The second one goes to the Todd's question about whether the LNP is still useful. And following Paul's comment, I think LNPs are still useful for the possibly extrahepatic by combining an extra ligand and where the GalNAc is not able to go now. And so those things, in principle, you know, Peter Cullis published a paper last month targeting the PSMA by taking a small molecule ligand using an LNP. So such targets are accessible. So I would say to my friend Todd, don't give up the LNP yet. I mean, you can still explore.
Thank you, Manu.
Thank you.
Yes, Raj?
I have a question for Mark. So in your presentation, you mentioned that in new process, using 1 micromole since 1 millimole since you are achieving 4+ gram of oligo. The question is what chemical modification you use? And second one, beside using polystyrene support, what other you change in the method? Because historically, people get 1 millimole scale around 3 to 3.5 gram purified oligo. Mm-hmm. So what advancement happened that way?
So in this specific project, I think the most important contribution to the increase in yield is the fact that the synthesis of all those strands has been made on this polymeric support, and in this specific case, the nitto phase. That, in my experience, has provided very good results for a lot of different projects. Modifications, those oligonucleotides are very simple, straightforward, first generation siRNAs. The backbone is all 2' it's all 2' hydroxy. So TBDMS synthesis and 2 steps cleavage and deprotection with removal of TBDMS. And there's no 2' methyl, no 2' fluoro, not even a single phosphorothioate in those oligonucleotides. And we got this pretty high yield anyway. I hope this answers your question.
Okay.
Yeah.
Thank
you. Thanks for the workshop. It's been very informative. Just wanted to elaborate a little bit more on it's really nice to hear people talking about raw materials, and Mark, it's great to hear you saying don't use more modifications than you need to. It can be very difficult if R&D groups kind of run wild and put in all their latest pet project modifications into a molecule. It can make things quite difficult to then take forward through clinical development. I just want to broaden out into supply chain and just say, if you go to one of the major CMOs and you're using sort of stock amidites like we've been talking about, 2' fluoros, 2' methyls, that's fine. Those manufacturers will supply those to you. Behind the scenes, you won't know about it. You don't have to worry about it in a large sense at phase 1. If you're using your modification of the week, as it were, in your molecule, that's now on you as the sponsor, and you'll be the one running the supply chain, going off to wherever it is in the world to get that material made and be responsible for it in terms of its regulatory status, BSE, TSE, also in terms of its performance. If it fails in the manufacturing suite, that's now on you in most cases. So just kind of want to warn people to just be very cautious about going off and sticking a bunch of modifications into something unless you really, really need them. If it makes it work, sure, put them in, but you have to accept the consequences in terms of you're now responsible for it going forward. Whereas if you don't need it and you put it in, now you're carrying that burden for more. So sorry, there wasn't a question there. It was more of a diatribe.
Yeah, it's correct.
Is anybody else? I also want to make a comment in addition to what Mana, my old friend, had made. So 2' fluoropurines, in fact, A and G, are actually toxic compounds at very high doses. As a triphosphate, they do hit mitochondrial polymerases and other human polymerases. The fact that you can use them safely in oligonucleotides is a really threshold effect. Your oligonucleotides are very potent. The amount of free triphosphate which can come from degradation of these 2' monomers is very low. Therefore, they are safe. But that doesn't mean that you really have to, indulge in using on 2' fluoros. That's, known fact. 2' fluoroadenosine was a compound very active as antiviral, but its development has been stopped because of toxicity. Just a comment.
Thank you, Leo. Okay. We are a little bit above schedule, so it's time, I think, to stop and have some lunch. Thank you very much, and see you back in the beginning of the afternoon for the real start of the meeting. Thank you very much. Enjoy your lunch.